Welcome to our journey into the fascinating world of cellular biology. Today, we're exploring the essential processes that allow substances to move across the cell membrane. The cell membrane, also known as the plasma membrane, is a dynamic and selective barrier that controls the movement of substances into and out of the cell. There are several mechanisms for this transport, broadly categorized into passive and active transport. Let's start with passive transport, which does not require energy. Molecules move down their concentration gradient in this type of transport. First, we have simple diffusion. Here, small or lipid-soluble molecules move directly through the lipid bilayer from an area of higher concentration to lower concentration. Next, facilitated diffusion. This process involves protein channels or carriers that help larger or polar molecules cross the membrane, again moving from higher to lower concentration. Osmosis is a special type of passive transport where water molecules move across a selectively permeable membrane from lower to higher solute concentration. Active transport, on the other hand, requires energy, usually in the form of ATP, to move substances against their concentration gradient. In primary active transport, ATP directly powers the movement. A prime example is the sodium-potassium pump, which maintains the electrochemical gradient by moving sodium out and potassium into the cell. Secondary active transport uses the energy from one molecule's gradient to transport another molecule. In Symport, both molecules move in the same direction, like glucose and sodium co-transport. In antiport, they move in opposite directions, such as in the sodium-calcium exchanger. Vesicular transport handles the movement of large particles or macromolecules, and it also requires energy. Endocytosis involves taking substances into the cell. In phagocytosis, or cell eating, large particles are ingested. Pinocytosis, or cell drinking, takes in fluids and dissolved substances. Receptor-mediated endocytosis is highly specific, allowing cells to internalize particular molecules after they bind to receptors. Exocytosis is the process of expelling substances from the cell. Vesicles fuse with the plasma membrane to release their contents outside, such as neurotransmitters from neurons. Transport proteins play a crucial role in these processes. Channel proteins form pores that allow specific molecules or ions to pass through. These channels can be gated, opening or closing in response to stimuli. Carrier proteins bind to molecules and change shape to shuttle them across the membrane. An example is the glucose transporter, GLUT. Pumps, like proton pumps in the stomach lining, use energy to transport molecules against their gradient. Understanding these transport mechanisms is vital for grasping how cells interact with their environment, take in nutrients, expel waste, and maintain homeostasis. Thank you for joining us on this exploration of cellular transport. Be sure to like, share, and subscribe for more deep dives into the wonders of biology.